This is the third and the last video on the gastrointestinal tract. In it, we're going to focus primarily on digestion and absorption of nutrients. And at the very end, we'll take a quick look at both anatomy and function of the large intestine. And we're going to start by talking about the digestion and absorption of carbohydrate. And two of the primary forms of carbohydrate are both starches, amylose. Notice again, we've talked about this before, especially last semester, but the carbohydrate ends in OSE. And then a related one is amylopectin. You can see that both amylose and amylopectin are just a string of glucose molecules that are linked together. Okay, the difference being that amylose is primarily just a straight chain, whereas amylopectin is branched. You know that we do begin some carbohydrate digestion in the mouth because that's where we first encounter salivary amylase. And that continues to a small extent in the stomach, but the amylase is quickly inactivated as it's exposed to the very low pH of the stomach. And then as we move into the small intestine, then the chyme will encounter the pancreatic amylase and we will continue further digestion of the carbohydrate. Again, these are hydrolysis reactions and we are breaking bonds until ultimately we get down to the disaccharide maltose, and again OSE as opposed to ASE. And also maltotriose in some cases, which is a trisaccharide, and then you can see alpha-dextrins. But in either event, again, these are individual glucose molecules that are linked together, and we're going to break those apart. Here again, you can see action of pancreatic amylase being the enzyme. And so each one of these is a glucose molecule, and this is starch, so more than likely it would be amylopectin because it's branched, and not amylose because it's not a straight chain. But again, you can see that we break this down primarily to the trisaccharide and the disaccharide. And then here you can see dietary carbohydrate, and we're going to work our way down from the mouth to the stomach and into the small intestine. What is lactose? What is sucrose? You might pause the video and see if you know what that is. Lactose is a disaccharide and it's milk sugar. And so it's the sugar that's found in dairy products, milk, cheese, yogurt, things like that. Sucrose is table sugar, and it too is a disaccharide, and it's made up of glucose and fructose that are linked together. And then the other disaccharide is maltose that we've already looked at. And so the lactose and the sucrose make their way all the way from the mouth through the esophagus and the stomach and into the small intestine, and they're not acted on by amylase at all. Once we get to this point, we get to the disaccharides, amylase has done its job, and we're all finished with it. But we're not done digesting these. And so now you can see here that lactose is further digested by lactase. This represents the brush border of those enterocytes, and so here are those microvilli, and you can see that lactase and maltase and sucrase are enzymes that are in the phospholipid bilayer, and so lactase will act on lactose to break it down to galactose and glucose. Maltase acts on maltose to break it down to glucose, and finally sucrase will act on sucrose to break it down to glucose and fructose. And so again, lactase, maltase, and sucrase are all other hydrolytic enzymes, but they're not in the lumen, they're in the phospholipid bilayer of those epithelial cells. The glucose and the galactose are absorbed by secondary active transport using the sodium glucose linked transporters, and here they are. And so you can see sodium is coming in, going down its concentration gradient, and as it does that, it will pull glucose and galactose along with it, and so galactose and glucose come along. There might be a little bit that occurs via facilitated diffusion using a GLUT molecule. Fructose is absorbed using that method, facilitated diffusion using a GLUT molecule. And so you can see that the fructose gets across and into the epithelial cell by a slightly different method than the glucose and galactose. Okay, and so now once we're in here, now we use GLUT molecules to get those across. and move them into the interstitial fluid, and then they get picked up by that capillary, which is in the middle of the villus. And so there it is. And now you can see that all three monosaccharides, galactose, glucose, and fructose, use the GLUT molecules. I don't know why they put them here. They should be embedded within the phospholipid bilayer. And they use the GLUT molecule to get across via facilitated diffusion. And then again, they've done a good job of putting the sodium-potassium pump here to remind you sodium has to be pumped out so it can continue to move down its concentration gradient. And that is an active process, and so it requires the use of ATP. But again, the pump is in the phospholipid bilayer. It's not in the cytoplasm close to the phospholipid bilayer. And then again, those are GLUT molecules. I don't care that you know the number. I should just take it off, but those are GLUT molecules. And here's just a different depiction showing you what we just talked about. And so again, glucose and galactose come across using co-transport with the sodium, and fructose comes across using facilitated diffusion and a GLUT molecule. This does not show you those hydrolytic enzymes that are found in the membrane to break down the disaccharides, so don't forget about those. And then again, all three monosaccharides will use a GLUT to get across into the interstitial fluid, and then they get picked up by the blood in that arteriole that's in the middle of the villus. Now we're going to start to look at the digestion of proteins. And again, digestion of protein did begin in the stomach. Remember, pH of the stomach is really low. It's a pH of 2 or lower than that. And you know that when you put a protein into a very low pH, it gets denatured. That changes its shape, and it helps the proteolytic enzymes attack the peptide bonds and break this thing down. And you know that some protein digestion occurred in the stomach using the proteolytic enzyme pepsin. Now we get here into the lumen, and the pancreas has added trypsinogen, chymotrypsinin, and procarboxypeptidase, and those are not yet proteolytic enzymes. We need to activate them. And that is started, again, using enzymes that are embedded within the phospholipid bilayer. And so the maltase, sucrase, and the lactase are embedded in the membrane, similar to the way you see these enzymes. And so the trypsinogen is cleaved to make it into trypsin, and then the trypsin will cleave the chymotrypsinogen and the procarboxypeptidase to make them into their active enzymes, as opposed to the inactive zymogens, which they are when they leave the pancreas and first enter the small intestine. And again, these are referred to as enteropeptidases because they're in the cell membrane of the enterocytes. And so here again, we can see here are some proteins that are being acted upon by the proteolytic enzymes. And they're going to break them down into individual amino acids and perhaps small peptides. And so dipeptides in some cases can in fact be absorbed, but most absorption is of the individual amino acids. And so we've broken all of the peptide bonds. Don't forget, a peptide bond is just simply a special covalent bond. And then most of the amino acids are going to use sodium cotransport the way that glucose and galactose did to move across the cell membrane. And we have several different transporters for the different classes of amino acids. Some research has looked at increasing water transport by adding certain amino acids to carbohydrate electrolyte beverages to accelerate rehydration, but to this point, it really hasn't worked any better than just having sodium in a drink that you might take. And so here you can see the co-transport, and they're just showing it to you in general. Not all amino acids use the same transporter, but this is co-transport, and sodium is going down its concentration gradient and dragging amino acids with it. And here you can see that some small peptides, dipeptides, perhaps tripeptides, can actually use co-transport with a proton coming in. And then you see we get that proton back out using antiport with the sodium. And then again, those amino acids will move across via facilitated diffusion, and then they get picked up by this arteriole that is in the villus. Now we're going to talk about the digestion and absorption of fats, and this is different than digestion and absorption of both the carbohydrate and the protein, and that's because, again, fats are not water soluble. They are not polar, and so they don't dissolve in the water very well. But the action of the stomach and the small intestine are trying to break the fat globule down into individual droplets. Now, to prevent these droplets from coming back together to reform that big globule, we use the bile salts. That's where they come in. And these bile salts are special. Here is one up here, and you look at it, and you should tell me this looks like it's cholesterol. And the bile salts are indeed made from cholesterol. That's lipid. It's a lipid, it's nonpolar, and so it's going to be hydrophobic. Well, here you can see we've got a polar part. This is going to be hydrophilic, and so the bile salts are amphipathic, much like phospholipids. And these bile salts will adhere to the lipid droplets, and that helps to prevent them from coalescing to forming those big fat globules again. Here's that nonpolar fat globule, and through the action of the stomach and the small intestine, it gets broken down to fat droplets, and then the bile salts quickly adhere to them. This actually makes the job of the lipase easier. Now the lipase can get in there and attack the bonds between the fatty acids and the glycerol a little bit more readily. And generally what we do is we break this down into monoglycerides, a few diglycerides, and then also free fatty acids, and those get packaged into what are called micelles. And so that's the importance of these bile salts that are delivered via the bile. And again, remember the bile salts were made in the liver. And so here's another place where cholesterol is important to you, although as it turns out, it isn't essential. Not essential. And now we see a review of what we just talked about. Here's that large droplet or globule, and it gets emulsified by the action of the stomach and the small intestine. And then in the small intestine, it quickly gets surrounded by the bile salts to prevent these droplets from coming back together and coalescing into that big fat droplet again. And then the lipase, in this case now it's pancreatic lipase, is attacking it, forming mostly monoglycerides, but some diglycerides. And then over here, here's the micelle, and you can see the components of it. And so here you can see monoglycerides, but you can also see some diglycerides as well. And then you can see the phospholipids. Oh, okay. And you can see fatty acids, which are primarily on the interior. You can see cholesterol primarily on the interior. And then the bile salts that help it to associate with water a little bit better than it otherwise would. And so here again, we can see a little bit of a schematic of that digestion of the triglycerides. And so there we go. We get monoglycerides and some free fatty acids. And so that's what's happening here in the lumen of the small intestine. And so now you've seen the importance of the bile salts as well as the lipases. And then here's that micelle that we just talked about. And you can see it moves down in between the microvilli of the enterocytes. And now because the components of this are they're nonpolar, they can move across the phospholipid bilayer of the enterocyte and get here into the inside. And so that's what happens. They move across via diffusion, and they get into the inside of the enterocyte. That's what this is. This is an enterocyte. And so again, small intestine is a simple columnar epithelium with lots and lots of microvilli. Once we get back inside, we kind of recombine this stuff to get triglycerides, and we package it up with protein and other stuff to create chylomicrons. And then these chylomicrons are released on the basal side via exocytosis. And so again, these chylomicrons are triglycerides, protein, and also a fair amount of cholesterol. So they're a little bit like low density and high density lipoproteins, but they're slightly different. And so they're referred to as chylomicrons. Here you can see a chylomicron, and you can see the components of it. It's mostly triglyceride on the inside. And then it's some cholesterol on the inside, and then the outside is phospholipid and protein, much like the membrane of a cell, but now it's not a phospholipid bilayer, look, it's a single phospholipid layer. not a bilayer. And so that's what's actually moving across over here. And it's going to get picked up by the lacteal. And the lacteal is part of what? Yes, it's part of the lymphatic system. And again, we find it up in that villus. That's where it's located. And so just as a summary, here is an enterocyte, and here are the microvilli, and we form the micelle, and the micelles move down in between the microvilli, and then again, by passive absorption, the components, the fatty acids, the monoglycerides, the cholesterol, they're all nonpolar. This is a phospholipid bilayer. It's mostly nonpolar, and so those things can move across. Here, I guess this is supposed to be their representation of the smooth endoplasmic reticulum, and we recreate the triglycerides. Again, as we've said, most of the fat both in your diet and in your body is in the shape of triglyceride. And then we package that up, triglycerides, protein, cholesterol, to create this chylomicron, and then it's released via exocytosis, moves into the lacteal, and that will be eventually transported back to the cardiovascular system. And even this stuff will make its way to the liver, where it will be processed post-digestion and absorption. And now we move on and we finish up with the large intestine. We're going to look a little bit at it. And so here you can see that the large intestine meets up with the small intestine at this part referred to as the cecum. And so that's this blind-ended tube. Connected to it is the appendix, which does seem to play an important role. It has a whole bunch of the good bacteria that are important here. And it also is a part of your lymphatic system. And so it does help with immunity. And then here we have the ascending colon. That's because the material now is moving up. We're sucking water out of it and it's becoming drier. Then we have the transverse colon because it is primarily going across. The descending colon. And finally, the sigmoid colon, because, if you twist it the right way and if you're smoking a little bit of weed, it does look like it's kind of S-shaped. And so that's why it's called the sigmoid colon. And then just to review real quickly, here's the transverse mesocolon, and that's going to attach the transverse colon to the posterior wall of the abdominal cavity. And then here is the sigmoid colon as well. And so there you can see that. And then again, a specialization here is the longitudinal smooth muscle is this band of stuff right here on the outside, and it's called tenea coli. And so that's the longitudinal smooth muscle of this thing. Well, most digestion by now has been completed, and as has been most absorption. But the large intestine is able to continue to absorb water and some salt as well. There is this huge population of bacteria, which for the most part are good for you. These are important bacteria, and they're very helpful to you. They actually will do some digestion of fiber, which you can't digest. You don't have the enzymes that are required to break down fiber, but some of these bacteria do. And this is good. As a result, these bacteria are helping to produce two vitamins, vitamin K and biotin, one of the B vitamins. It's also producing these short-chain fatty acids, which don't seem to be important much of any place except here in the colonocytes. They're actually used as the energy source for the colonocytes, and these short-chain fatty acids also seem to offer you some protection from colon cancer. And so they are indeed good for you. They are indeed good for you. Another thing that you can do if you don't want to have colorectal cancer is cut way back on your beef consumption, any red meat for that matter, any processed meat. And so things like pepperoni and salami and all of that kind of stuff. And so all of those things do and can cause colorectal cancer. And there is a little bit of data that suggests that colonocytes actually have amino acid transporters. And so we may come to find out that a little bit of amino acid absorption can also occur here in the large intestine and not just in the small intestine. And then finally, we'll look at a little bit of mineral absorption, but we're not going to worry too much about this. Most of the sodium absorption is occurring using the SGLT1s. And so again, that's helping with absorption of other stuff like glucose and galactose and amino acids. Sodium absorption can be influenced by the type of carbohydrate that gets put into a glucose electrolyte solution that gets marketed to athletes and people who exercise in the heat and humidity. Chloride and potassium, they actually can be absorbed through paracellular channels in between the cells, much like what we talked about for the kidney with reabsorption. Calcium, iron, and zinc also use solute-specific transport mechanisms, and so the sodium can be helpful there in absorbing those. And then again, we're always trying to study what might help with absorption of things. There's no evidence that water or solute absorption is enhanced by addition of any common mineral other than sodium chloride. And so that brings us to the end of digestion.